Hello everyone, welcome to MMBS-111 Introductory Microbiology. This is our final mini lecture from Chapter 5, and we'll be talking about a number of different components here, including tissue culture, other non-cellular structures that cause disease, and then how are scientists or how's medicine trying to address viruses. There are 3 learning objectives for this mini lecture. First, know the difference between the terms in vitro and in vivo. List some other infectious agents, and be able to answer the question, how are scientists trying to target viruses? So let's look at these terms. We're not going to cover a whole lot from section 5.4, but there are some things that you need to know. Okay? Viruses require living cells as their growth medium. So outside of infecting actual living things with viruses, it was very difficult to study viruses and viral diseases until the development of tissue culture. So we're able to study viruses in 2 ways. In vivo, which means within the actual organism. And when we talk about in vivo virus studies, we're generally looking at embryonic tissues like bird eggs. So in vivo means within the organism. In vitro is outside the organism and generally means within the laboratory. And when we study viruses in vitro, we're looking at viruses that have infected cells in a cell or tissue culture, and these have all been developed and grown in the laboratory. There are a number of reasons that we want to be able to grow and study viruses outside of their hosts, and that is to be able to isolate them and identify them so that we can better understand them in a clinical setting, be able to use those viruses and prepare them so that we can develop vaccines, and also be able to do research on how viruses replicate, what are the cycles, understand viral genetics, and how viruses affect their host cells. So the development of cell and tissue culture was a really incredible development in science and has allowed us to study a number of things that we could not have done otherwise. In a tissue culture, in a cell culture, animal cells have been isolated, and then they've been provided with a medium, a growth medium, that contains all of the necessary nutrients and other compounds that the cell needs in order to survive outside of the body. Cell cultures are grown in sterile chambers to prevent contamination, and the cells don't grow in complex tissues like we see in a body, but they actually form a single layer that we call a monolayer. And this allows scientists and researchers to really see the cells and be able to inspect those cells for any types of infection, especially when we're studying viruses. There are a number of other infectious agents that are not cellular and not virus either. One of those is prion, and prions are proteins. They do not include any nucleic acids. They are implicated in a number of chronic and persistent diseases in humans as well as animals, and there are some examples there on the left hand of your screen. That is normal brain tissue up at the top, and then the others are example of brain tissue that has been infected by prions in a number of different brains. So you have scrapie, which is a prion disease that infects sheep brains. You have Creutzfeldt, Jakob, and Kuru, which are both diseases of the human brain that are caused by prions. These diseases are progressive and universally fatal. There is no cure for spongiform encephalopathies. They tend to be latent, so they are dormant for a very long period of time before any symptoms arise in the host. And there are a number of examples provided there at the bottom. Some we've already mentioned here in the images. Chronic wasting disease, if you're a hunter, you probably heard of that in deer and elk. Also, mad cow disease is bovine spongiform encephalopathy. And all of these diseases are caused by prions. Another non-virus or non-bacterium infectious agent are these satellite viruses. And they're not a true virus because they can't function on their own, but they are dependent on other viruses for replication. So typically a satellite virus is only able to infect a cell that has already been infected by another virus. Some examples are given here on the slide. Adeno-associated virus. It's named adeno-associated virus because it was originally thought that it could only infect other viruses that were infected with the adenovirus. That has since been discovered to be incorrect. It can infect other cells that have been infected by other viruses. Delta agent, it's an interesting name, is just a small circle of RNA, and it sort of accompanies hepatitis B, so it can only be expressed when hepatitis B virus is present within the cell, and it actually increases the severity of hepatitis B liver damage. The last non-virus is called a viroid, and these actually don't cause any diseases in animals, but they are more important as far as plant diseases and infections. They are similar to viruses, but they are much smaller. They are composed of naked strands of RNA. They don't have any capsid or other type of external coating that protects their nucleic acid. And they are significant pathogens to a number of agricultural products and can cause a number of problems economically for farmers. So viruses have a pretty significant impact on human health. They are common causes of a number of acute infections like colds, chickenpox, hepatitis, influenza. They are also prominent worldwide. There are a number of severe viral infections, including dengue fever and Rift Valley fever, yellow fever. They cause infections with high mortality rates like AIDS and Ebola. They also can cause long-term disability. So they're associated, or they are the causative agents of polio and neonatal rubella that result in long-term disability of the host. And they're also associated with chronic infections like MS, cancer, type 1 diabetes, Alzheimer's, and even obesity. So why don't antibiotics affect viruses? We've developed antibiotics to treat disease, yet they aren't effective on viruses. There really is no point to taking an antibiotic when you have a viral infection. And the reason is because antibiotics are developed to target certain components of bacterial infections, including the cell wall components, cell wall synthesis, cell membrane synthesis, and metabolic pathways. none of these are possessed by viruses. So taking an antibiotic is not going to have any effect on the virus. And since viruses have to hijack the host cells in order to reproduce, to be able to inactivate a virus, you must also kill the host cells. And this can result in some severe side effects. Most antiviral drugs have been designed to block viral replication, and they do this by targeting the function of the host cells being infected by the virus. And this can lead to a number of pretty detrimental side effects. Draco, double-stranded RNA-activated capsase oligomizer, was developed a number of years ago, and it is designed to cause any cell within the host to destroy themselves if they are infected with the virus. The problem is that it doesn't matter what the virus is. So you could have a latent virus hanging out in your cells, like herpes or varicella, that isn't causing any issue for you right now, but maybe you have an active virus. This Draco would cause any cell within the host to destroy themselves, whether the virus is currently active or latent. The best way to prevent a viral infection is through vaccination. Vaccines are very valuable and have been proven time and time again to prevent the spread of disease. Polio has been eradicated in a number of locations. Smallpox has been eradicated worldwide because of the use of vaccines. There is, unfortunately, a limited number of vaccines that have been developed to address certain viruses. So not every virus can be prevented through vaccination. But vaccines have definitely been proven to be very effective against those that have been developed. Okay, so that is the end of Chapter 5. Make sure that you go through these questions here. Be sure that you can answer each one correctly. Go back and review the objectives. And hopefully you found this video helpful.